Hello viewers, hope that all are fine. I am Dr. Sharif Hussain. Today my lecture or presentation topic is antioxidant, carbohydrate and protein in fruits and vegetables. It is under the courses of nutritional science or nutritional biotechnology. Let's see here some fruits. This is dead fruits. This is ajwa dates, and this is the dates, fresh dates, sukari, and this is water apple or wax apple, and this is the olive fruits. This is the vegetables, okra or lettuce finger. This is the medicinal plants, pigments plants like bougainvillea, and this is I published this book, Antioxidant in fruits and vegetables, carbohydrates and proteins. Okay, let's move to next topic. Details title: Introduction to nutritive fruits and vegetable productions, nutritional biotechnology, antioxidant, phytochemicals, food and human nutrition. It is so. Let's go to define nutrition. Or what is nutrition? The branch of science that deals with nutrients and the nutritions, particularly in humans. It is the process of providing or obtaining the food necessary for health and growth. In other senses, nutrition is the science that interprets the interaction of nutrients and other substances in food in relation to maintenance, growth, reproduction, health and disease of an organisms. It includes food intake, absorptions, assimilations, biosynthesis, catabolisms, and excretions. Next we can see human nutrition. Human nutrition refers to the provisions of essential nutrients necessary to support human life and health. Generally, people can survive up to 40 days without foods. A period, a period would last largely depending on the amount of water consumed, stored body fat, muscles, mass, and genetic factors. Plant biotechnology develops the essential food nutrients and its quality to humans and livestock health, including carbohydrate, lipids, proteins, minerals, vitamins, directly or indirectly. Then we can define food nutrition. What is food nutrition? Food nutrition is the nourishment by energy that is obtained from food consumed or the process of consuming the proper amount of nourishment and energy. An example of nutrition is the nutrient found in fruits and vegetables. Also, the example of nutrition is eating a healthy diets. Plant breedings and biotechnology based techniques are needed to improve nutritional value with the desired quality traits in different fruits and vegetables. So plant biotechnology based techniques or technology most important to improve the nutritional value in different fruits and vegetables. So it is correlated, plant biotechnology and nutritional science and nutritional biotechnology. It is correlated. Continuing improvements in molecular and genomics technologies are contributing to the acceleration of nutritional products, development for human health, benefit and nutrition. Then we can see or we can define phytochemicals. What is phytochemicals? From Greek, phyto means plant and chemicals means chemical produced by the plants. So phytochemicals are chemicals produced by plants through primary or secondary metabolisms. This is organic chemicals. This is organic substances or chemicals. Phytochemicals under research can be classified into major categories such as carotenoids and polyphenols, which include phenolic acid, flavonoid, anthocyanin, stilbenyl lignans. Chlorophyll is a class of phytochemicals that is responsible for giving plants their green pigments. Chlorophyll, carotene or carotenoid, this is one kind of xanthophyll, one kind of pigments. Studies have found that chlorophyll has anti-inflammatory, antioxidant and wound healing properties. Anti-inflammatory, antioxidant and wound healing properties. List of plants containing phytochemicals or plant chemicals like vegetables, broccoli, tomato, okra. This is most important vegetables and bearing lot of pigments. Broccoli usually pigments and green pigments. This is xanthophyll, like red pigments, okra also green pigments, and vitamin C and antioxidant contains a lot of antioxidants. Fruits and nuts and like almond. Yeah, common herbs, aloe vera, American ginseng, crown, ivy, hawthorn, hops. This is common is aloe vera. And the other fruits and nuts, almond. You can see here blackberry, blueberry, grapes, hazelnuts, mangosteen, olives, orange, pomegranate, red raspberry, strawberry. It contains a lot of vitamin C, antioxidant, flavonoids. And then here also you can see herbs other than ginseng, lemon balm, lemon verbena, marigold, red clover, the sweet clover, tea, carrots. These also different types of beans and seeds, cacao and soybeans. And also here we can see medicinal plants, different types of plants like long oat, opium, poppy, passion fruits, red, brownie, hibiscus flower, bougainvillea flower. So this is the medicinal plants. They contains a lot of antioxidant. This antioxidant is most important for our for the human nutrition and prevent the diseases. So let's go to discuss antioxidant. What is antioxidant? Antioxidant are the phytochemicals. Basically, it is phytochemicals from plant sources, vitamins and other nutrients. There is phytochemicals, vitamins and other nutrients that protect our cells from damage caused by free radicals. It contains by its free radicals, it protect or prevent our cell from damage. So this is the most important antioxidant. In vitro in and in vivo studies. New studies have shown that antioxidants help prevent the free radicals damage that is associated with cancer and heart disease. They have free radical and damaged. When free radical prevent the free radical damage or cell damage that is associated with cancer and heart disease. Antioxidant can be found in most fruits and vegetables, but also culinary herbs and medicinal herbs can contain high levels of antioxidants. Draglands and colleagues, their scientists, showed in their studies that several culinary and medicinal herbs are important sources of dietary antioxidant, published in Journal of Nutrition, 2003. The antioxidant level of herbs can be as high as 465 millimole per 100 gram. They, they found a good antioxidant level for 465. A study in 2006 by Thompson showed that a botanical diversity of fruits and vegetables plays a role in the biological effect of antioxidant phytochemicals. Consumption of smaller quantities of many phytochemicals may result in more health benefits than the consumption of large quantities of fewer phytochemicals. So these phytochemicals is most important to control the diseases. Functional mechanism of antioxidant and free radicals. Now we can discuss about the functional mechanisms of antioxidant and free radicals. Free radicals are formed as part of our natural metabolisms, part also by environmental factors including smoking, pesticide, pollutions and radiations. Free radicals are unstable molecules which react easily with essential molecules of our body, including DNA, fats and proteins. All organic and inorganic materials consist of atoms which can be bound together to form molecules. When a free radical attacks a molecule, it will then become a free radical itself, causing a chain reaction which can result in the destruction of a cell. Antioxidants have the property to neutralize free radicals without becoming a free radicals themselves. When antioxidants neutralize free radicals by receiving or donating an electron, they don't become antioxidants themselves because they are stable in both forms. Well-known example of antioxidants are the vitamin C, E, and beta carotene. So beta carotene, vitamin C, E, this is also antioxidant. So we also determined vitamin C, beta carotene in case of many fruits and vegetables and got very good result. Benefits of antioxidants. Numerous studies with plant phytochemicals, means antioxidant, show that phytochemicals with antioxidant activity reduce risks of cancer and improve heart health. Antioxidants reduce the risk of cancer. Not all results are inclusive, but many studies show that antioxidants may reduce the risk of cancer. A large randomized study on an antioxidant and cancer risk was the Chinese Cancer Prevention Study, 1993. This study showed that a combination of the antioxidant, beta-carotene, vitamin E, and selenium significantly reduced incidence of cancer. However, alpha tocopherol, beta-carotene cancer prevention study showed that intake of beta-carotene increased lung cancer rate of male smoker. Let's move to antioxidant protect the heart. Other topics. Everyone knows that cholesterol causes heart diseases and tries to limit cholesterol intake. But a more important causes of fatty buildups in the arteries in the oxidations of low density lipoprotein cholesterols. The use of the dietary supplements of antioxidant could reduce the risk of the cardiovascular disease, but there is no hard hard evidence. At this stage, studies only show that the intake of foods naturally rich in antioxidant reduce this risk. Now we are discussing regarding biochemical content in plants and biochemistry and biological chemistry. Biochemical contents. The science deals with the chemistry of living matters, the chemicals, composition of particular living systems or biological substances. This is called biochemical contents. On the other hand, the study of the chemical substances, vital processes occurring in living organisms, the chemical compositions of a particular living system or biological substances. Biochemistry is the study of structure, compositions, and chemical structure of reaction of substances in living systems. And then biological chemistry, sometimes biochemistry, sometimes called biological chemistry, the study of chemical composition process within and relating to living organisms, the controlling information flow through biochemical signaling and the flow of chemical energy through metabolisms. Biochemical process give rise to the complexity of life. So this is correlated biochemical content, biochemistry, and biological chemistry. Example of biochemical contents like carbohydrate like sugars, glucose, fructose, sucrose, starch, starch, cellulose, fibers, sorbitals, pH, titratable acid, etc. Nutritive plant production or nutritional plant production. Nutritive or nutritional plant production is a process or method of the growing of plants or crops, either for sales or domestic consumption using different techniques and nutritions. The crops refers to the produce of plants that are cultivated, like according to the US EPA. Crop production is a complex business requiring many skills such as biology, agronomy, mechanics, and marketing, and covering a variety of operations throughout the years. Nutritive plants can be produced by in vitro and in vivo conditions. US Environmental Protection Agency, according to their statement, this year called US EPA, their statement, we mentioned this. Technology involved in developing nutritional value in plants for human health benefits. Different types of technologies. Uh, Using these technologies, we can develop nutritional value in plants for human health benefits. So, like plant biotechnology or nutritional biotechnology, directly related to develop nutritive value in plants for human health benefit. So this is correlated development of nutritional value in plants and human health benefits and plant biotechnology or the nutritional biotechnology. All are correlated, almost same field. So we can use the techniques or technology to improve the nutritional value for human health benefit, like micropropagation, one techniques. Micropropagation is widely used in agriculture or botany, forestry, and floriculture. Micropropagation can also be used to conserve rares or endangered plant species. It can be done from embryo, shoot, leaf, root, and tips. So by micropropagation, we can increase the nutritive value in plants. Then other techniques is liquid culture in bioreactor. By this culture, large-scale growth of plant cells in liquid culture are done in bioreactor for production of valuable compounds like plant-derived secondary metabolites and the recombinant proteins. The others is photoplast fusion techniques by which two photoplasts are fused to form the hybrid cell that can grow into mature hybrid organisms. Full plants usually performed on plants. Distantly related species are processed by photoplast fusions and generations of the novel hybrids can be done. The other techniques is transgenic plants productions. This also by this way we can increase the fruit nutrition. tDNA transformations can be done using tissue cell followed by either short-term testing of genetic construct or regenerations of the transgenic plants to increase the plant nutrition. Other techniques, meristem tip culture, also we can use these techniques. Certain techniques such as meristem tip culture can be used to produce clean plant materials from virus uh, stocks such as potatoes and many species of shop fruits. This is from various stocks. Various stocks. And then cell culture. Cell culture is a complex process by which cells are grown under controlled conditions generally outside of the natural environment. In practice, the term cell culture has come to refer to the culturing of the cell derived from multicellular eukaryotes, especially uh, plant cell, animal cells. However, there are also culture of plant cells, fungi, and microbes, including viruses, bacteria, and protists. So by this way also we can increase our nutritive value in plants or in the cell. In other topics, cell suspension also we can do it by cell suspension. Other topics, cell somatic embryogenesis. This is also one kind of techniques. Other techniques, somatic embryogenesis using the embryo and growth regulators, and then different types of cytokinins, and then auxin cytokinins by using this. So you can see here different many scientists they use this and they got the good result. We can see here class stages that allow shortening of the process of somatic embryogenesis and provide basis of morphological, biochemical, and molecular studies of the nature of somatic embryogenesis from single cells to whole plants. So it is cited. You can see there is some from these topics. So chapter 2 we can see here uh, plant nutrition enhance phytochemicals, food and human nutrition. So plant nutrition enhance the phytochemicals, food and human nutrition. So we can see a plant nutrition enhance the foods, fruits, vegetables, crops, grains and nuts. This is almost foods, different types of foods. Includes the food, fruits, vegetables, crops, grain, nuts. Contain almost all of the minerals and organic nutrients established as essential for human nutrition, as well as number of unique organic phytochemicals that have been connected to the development of good health. Like one thing is plant nutrition. Plant nutrition is the study of the chemical elements that are necessary for plant growth. There are several principles that's applied to plants. You can see here different types of the some scientists also said about the plant nutrition. Essential plant nutrients. 16 chemical elements are known to be important to the plant nutrient growth and survival. 16 chemicals elements. 12 the main groups, non-minerals and mineral groups. Non-mineral groups is like hydrogen, oxygen, and carbon. And mineral groups, others is mineral groups. These nutrients are found in the air and water. Or in a process called photosynthesis. Plant uses energy from the sun to exchange carbon dioxide, carbon and oxygen, and water, hydrogen and oxygen into starch and sugar. The starch and sugars are the plant's foods. Photosynthesis means making things this life. Since plants get carbon, hydrogen, oxygen from the air, water, there is little farmers and gardeners can do to control how much of these nutrients plants can use. The mineral nutrients, 13 mineral nutrients essential for plants, comes from the soils, dissolved in water, and absorbed through the plant's roots. This for the some primary nutrients are nitrogen, phosphorus, potassium. The major nutrients usually are taken from the soils first because plants use the large number of for their growth and survival. The secondary nutrients are calcium, magnesium, and sulfur. We can say something micronutrients. Micronutrients are those elements essential for plants growth which are needed is only very small quantities. This is called micronutrients. Like micronutrients according to soil science society of America, like boron, copper, iron, chloride, manganese, molybdenum, zinc is called is 7 macronutrients. And macronutrients is these nutrients that are tends to the less available in the soil with low pH. Micronutrient tends to be less available in soil with high pH. So we can see the roles of macronutrients like nitrogens. Nitrogen is a part of all living cells and is a necessary part of all proteins, enzymes and metabolic process involved in the synthesis and the transfer of the energy. Nitrogen is a part of the chlorophyll, the green pigments of the plants that is responsible for photosynthesis. So nitrogen is the most important, helps plants with rapid growth, increasing seeds and fruits productions, and improving the quality of leaves and the forest crops. So it means the nutrition, to increase the nutrition of the plants. Phosphorus also, like nitrogen, phosphorus is an essential part of the photosynthesis, involved in the form of all oils, sugars and starches. Helps the transformation of the solar energy into chemical energy for plant malnutrition. Potassium we can see here absorbed by plants in large amount than any other mineral elements except nitrogen. Then we can see calcium, essential part of the plant cell wall structure, provides for normal transport and retention of the elements as well as strength in plants. We can see a sulfur, essential for plant foods for production of proteins productions. Then others, micronutrients, borons, also use of nutrients and then regulates their nutrients. Copper, important for reproductive growth, like flowering and fruits, aids roots metabolism, helps the utilization of proteins. Chloride is found in soils, aids plant metabolisms. Iron, essential for the formation of chlorophyll. Source of iron are the soil iron sulfate and chelate. Manganese also functions with enzyme systems involved in the breakdown of the carbon dioxide and the nitrogen. Molybdenum helps in use of the nitrogens. Zinc, essential for the transformation of the carbohydrate. So this is the all about the plant nutrition. This is important for the for the for development of the flowering leaves, shoot, then flowering and the fruits, and any good vegetables also. Then we can see here chapter, other chapter, phytochemicals, pigments and antioxidant properties in the bougainvillea plant. Flowers. Bougainvillea are the popular ornamental plants and used as official flowers in most areas with warm climates, including Australia, India, Malaysia, Mediterranean regions, Mexico, and other countries. And then bougainvillea, this is also called medicinal plants. Some scientists studied, and also this we can see here different types of the flowers, and the excision and the senses of dropping of the flowers. Very good flowers in bougainvillea, very nice color pigments. So it contains it means to increase the base lives of the flowers and for phytochemicals like chlorophyll, carotene, and antioxidant. This is the most important flower. Chlorophyll and antioxidant. Sometimes we can use this carotenes and chlorophylls, this color of the food additives. Natural organic color or organic food additives. So we can see different parameters. Chlorophyll A, B, and the carotenoid determinations. We determine the chlorophyll A, B, carotenoid, and the total phenol determination. Total phenol content determined. This is bougainvillea glabra as varieties, and this is the foliage assay. I described the singleton and the rossi, and anthocyanin content determination. Anthocyanin content determined anthocyanin content. Total anthocyanin and the carotenoid contents of the hydrophilic extractions on lizard using the pH differentiation methods with cyanidins, 3 glucosides used as a standard. Then we use the total antioxidant investigations by Prolox standard solutions, using Prolox standard solution, and then we use this samples, and then different types of molecules, TAC tags molecules, and then we use the distilled water, and after that we use the spectrophotometer. This is called the for absorbance at 570 nm. So this is total flavonoid investigations. Flavonoid we investigated aluminum chloride, chloromatic acid. You can see the controls, only two and this is the sucrose treated flower, many flowers. This is very high color, many flowers, flowing stress, and the 100 ppm AOA. Oxalo oxyacetic acid treated flower. So this is the stress flower. We got the high pigments. So now we can see some discussion of the result. Here chlorophyll A, chlorophyll B, and the carotene content. So in the control we can see here in the control it is less than in the treatments. Treatments got higher contents than the control one. So you can see here also the first anthocyanin, total phenol, flavonoid, and usually it is higher in the treated plants than control. Then others total phenols, controls here discussion and total antioxidant content already discussion we discussed. Then here we can see uh, chapter 4, antioxidant of water apple or wax apple fruits and nutritional value development by gibberellic acid hormone or plant hormone. This is the other topic. We can see introductions. Water apples is the common fruits in Malaysia as well as other Asian countries. The fruits is widely cultivated and grown throughout the Malaysia mainly as well as scale gender ranging from 1 to 5 hectare. Gardener skill gardener skill gardener ranging from 1 to 5 hectare with hectares average estimated on 1,500 hectare. So 2005 Jane Hong Shu at 2006. Actually, my PhD student did this experiment in Malaysia. That's why he mentioned the Malaysian the data in respect to Malaysia. So the food development and ripening have been considered as the most important phenomena in food productions. Then usually here he developed on usually people are doing the spraying of the spray and found better effect in English foods. But he innovated the techniques, swabbing techniques methods and small scale, micro scale, small scales, and then he developed spray drift, was caused by spray and the drift method. Also, Hosan developed the swabbing techniques and resulted excessive flower in these plants. So he used this and then he got the results. The gibberellic acid has been shown to increase the fruit shares and the growth and the clementines, orange, and also others. Different scientific data. They gave the data and very good data. Then they increase the food farmers, soluble solid sugars, and also food growth stopped. And they got less food crops and then also others phenomena. And other scientists they mentioned who mentioned the flavonoids, flavanol and glyceride and anthocyanin contains a lot of anthocyanins, contains different types of buds, initiation, flower stage and bud stage. Chlorophyll contains determine the chlorophyll contains leaves of the branch was determined by mineral of SPA split meta. And then total soluble solids contents measured by the refractometer. You can see a fructose and the inverted sugar centrifuged, then collected juice. After that, measured by Apago digital hand refractometer, Tokyo, Japan. Then PS determination of the juice, and then potassium content determinations by the potassium meter. It's called potassium meter. And total phenol contents of water apple determined by using the Foley's assay. So this is colorimetry methods, and then these details we can see here. Spectrophotometer. He used the spectrophotometer to 765 nm, and this is the almost same the fallen. This is called the fallen standard methods, reagent methods. Total flavonoids content also he measured by the ammonium chloride colorimetric assay, and using different types of the reagents like methyl hydroxide, sodium nitrate, ammonium chloride, like other sodium hydroxide. So this is the different types of total anthocyanin content also determined by Rodriguez and the Sauna et al. This was Sauna et al. 2001 according to their standard. This is the anthocyanin content equations using their anthocyanin equations. And we can see the resultant discussions. TSS content was higher in the different concentration of J3 that control. pH content also lower, a little bit higher in lower means more acidic, the higher means less acidic, alkaline. So this is higher in the different concentrations of J3 that control. This is the fructose and inverted sugar also higher in different concentration of J3 that control. Here we can see maturity development color 94, 98, J360, highest maturity content, more color, the outer foods. Flavonoid content was more in the different concentration of J3 than control. And total phenol content, phenols also higher in J3 30, 60 and 90 then the control, but the highest total phenol was highest total phenol content was found in the J360. This is all about the potassium content determined on higher the treated fruits that control. Then here also J3 30, 60, 90 spat chlorophyll content was found higher in the treated fruits, treated branches that control. This is the anthocyanin content, maturity development anthocyanin content, correlations. Anthocyanin content, maturity content was increasing, then anthocyanin content is increasing. So this is called correlation. When anthocyanin maturity content decreasing, anthocyanin content also decreasing. So this is called the decreasing trend. We see the color, the highest in the control, and this is the GST 60 ppm milligram per liter. Very good color, good result. And the some are the pigments that give most fruits. They are red color, violets and the blue color. When there is more colorful, that's why we will choose the foods, colorful foods. So when you want to buy fruits from market, then it is the most choice of anyone to choose the fruits more colorful one with high pigments. These pigments includes the flavonoids and also the antioxidant. Flavonoids compositions are antioxidant. Mentioned by different many of the scientists. Potassium is also important for the roles of the activators of the many enzymes. Potassium content. Also, J3 applications, they found that foods significantly increased potassium content. This is all about the conclusion. Is possible we can believe that fructose, inverted sugar, and the potassium content are related with the fruit's weight and size. Are considered as effective the fruit's setting and high growth rates as well as color content. Color is important for the fruits. Color, it was determined that some techniques J3 had a tendency to increase the juice contents. Food setting developed in water apple could be regulated by the external applications of the J3. Replicacy 60 ppm improved the quality of the water apples fruits, increasing length and the width of the fruits as well as increasing the contents of the total sugar inside the fruits. So other topic we can see pigments of the hibiscus plants. Different types of pigments we can see here. The hibiscus flower, chlorophyll A, chlorophyll B, and the carotene content determined. Hibiscus flower, chlorophyll A, B content. Those are sciences for control from stress and the thinness and the hormone applications. Different types of the treatments. We can see carotene content is higher. The treated plant their control is also higher than control. Chlorophyll A is little bit lower than control. So this is the most important topics. Also you can see this plant is also very good red color. When color will be very good, and then carotene content will be good. It means antioxidant will be very good. The amount of chlorophyll B was the higher than the chlorophyll A in hibiscus rosacea flower. This was followed by the carotene contents, which showed the lowest content, but the results the control treatment showed the highest amount of chlorophyll A, while J3 treated flower contained highest amount of chlorophyll B and carotenoid. Chapter 6, we can see a nutrition quality. Antioxidant and the mineral contents. Development in okra using plant growth regulators. Different types of the okra. This is one kind of vegetables. Vegetables are rich in vitamins, minerals, and dietary fibers that play an important role in human body for human nutrition. This is important. Vegetables, fruits, are most important for the human nutrition. They are considered as the protecting foods as the consumptions avoiding many diseases. Okra is one of the most important popular vegetable crops in tropical and subtropical areas in Asia. So this is the many scientists, they say this contains such as vitamin C, AB complex, calcium, potassium, iron, other minerals. We also found in our research data it contains different types of minerals, vitamin C. And other scientists also, they mentioned it contains minerals, potassium, calcium, magnesium, also other minerals. We use the growth regulators, and then in the flower, and then we see the results. Total soluble solid vitamin C contents. My PhD student did this work in Malaysia, and then he submitted his thesis, and he got graduation from the University of Malaya by this work. Nutrient content, different types of nutrients, analyzed the different types of nutrient contents. You can see here what settings, and you can see here different types of the vitamins, vitamin C. The highest vitamin C got in the treated plant there, the control. Actually, this vitamin C is like antioxidant. This is okra plants, okra seeds and seedless okra we produced. And then the different data according to our results, also total soluble solids, sugars or others found in okra. And then it is the different types of the mineral contents, antioxidant, vitamin C, calcium, potassium, magnesium, ferrous, iron, sodiums, different types of contents. You can see here the result. Yeah, here also it contains good potassium, calcium, magnesium, ferrous, almost is higher in the concentrations, 25, 50, 100, then zero control and higher concentrations, 200 people. So this is all about the results about the vegetables and also others. Physiobiochemical contents, minerals, and the DNA quantifications, like protein contents of dates, fruits, applying residual effect of lights, different light penetrations. Date fruits are very rich in the fiber, fats, and proteins. Date fruits. That's why that they have a form of sugars that bears the body high level of the mobility, heat, energy, and they can be easily broken down in the body. So this is most important. Dates fruits contain many vitamins A, beta carotenes, B1, B2, B3, B6, and minerals very high. We also determined and we got the very good result. That's why this dates fruits is the most important foods. They also contain the sodium, potassium, calcium, magnesium, iron, sulfur, and USDA, United for United States. They did research data. They mentioned Sunlight is considered to have the significant role for the fruit's quality developments. Sunlight. When there is sunlight, enough sunlight, more sunlight, then photosynthesis will be happened good, and then after that sugar productions. Uh, so will be good. They found this they found this data, and then they measure moreover the fruits is the exposed sunlight were found to have more pigments than the unexposed sunlight and shade one. When the place is this shady area, this is the exposed sunlight area. This is mid sunlight area, little bit dark. This is more dark inside. Here, exposed sunlight area. So full sunlight, light shadow and deep shadow. So we can see different types of result there. We did this work in Saudi Arabia, then fructose content determination, glucose content determination, inverted sugar determinations, percent acidity content, different types of meters, lactate meters, calcium meters, and also glucose, inverted sugar, and fructose meters. We use this, and then different types of potassium, calcium meters. We use the Horiba potassium meters and others, calcium Horiba calcium meters. Yes. DNA isolations, we did a DNA isolation by the technique CTAD. We use the different types of volumes, and then you can see here we use the chloroform and isoamine alcohol, and then these techniques we use, you can see very easy. 0.8% agar gel use 99.2% area and 0.1 microliter of ethidium bromide. This is important to complete the DNA isolations. So then in details you can see here we use the different types of, then this is called electrophoresis, gel electrophoresis. This is the main topic, gel electrophoresis you use, and then a small DNA fragmentation migrated more rapidly to the gel matrix compared to the large fragment, so resulting in the molecules separation based on individual size, resulting molecule result. Here the result we can see discussed already. Here first season, second season, full sunlight is higher than the everything is high, flower content and the fruits state is higher, and the full sunlight, then shadow and dark shadow, and also they are called the measurement of the fruit maturity content higher for full sunlight. This is full sunlight A1, 2, this is in middle, and this is the shadow sunlight. This is less, this is called the full sunlight maturity content is higher. Then other two, shadow, light shadow, shadow sunlight. This is still green, still not ripened. This is little bit ripened, but it's full ripened due to sunlight. Also same full sunlight, light sunlight and the dark sunlight is still not ripened, green color, but there is full ripened. So this is we already mentioned here, different days, great days, different types of. It is 60 days. Grade different maturity grade 5, 4, 2. The same days we have 4, 3, 1. First season, second season. Here also first, second season. Fruit length and fruit weight is also higher in the full sunlight, then shadow and dark shadow. Then here we inverted sugar higher in the full sunlight, then shadow and dark shadow. Fructose also same, higher in full sunlight, treated fruits, then shadow and dark shadow. Here also remember DNA yield also deep shadow, light shadow, full shadow, and full shadow. DNA yield higher, first season, second season also. Then light shadow, deep shadow. You can see here DNA band represented, actually protein means represented by the DNA band. We can see the protein content. The first season DNA measurements. first, second, third, fourth. This is the dark one. This is light penetrated. This is standard. It will flow as standard. This is the dark. This is the light. The full light. So this is the topics. So nutrient content, different types of nitrates, potassium, calcium content, first season, second season, first season, second season. When in full sunlight it is higher, then the shadow and the light shadow or dark shadow. Here also you can see pH determination, sodium and pH determination also. Full sunlight is higher, then dark shadow and the light shadow. Chapter 8, Antioxidant nutrients value and the DNA effect of azure-dates. This is also this dates is called sukari dates, and this is azure-dates. Azure-dates. Dates are notably one of the high caloric containing fruits. They are greater source of minerals and vitamins and composed health benefits, having flavonoid, polyphenolic antioxidant known as tannins. So different types of this process anti-infective, anti-inflammatory, anti-hemorrhage, prevent easy bleeding tendency. So this is the Umesh 2009. He reported this. So it contains the vitamins and then beta carotenes, B1, B2, B3, B6. Ajwa dates are the best and medicinal and nutritional fruits compared to the other dates varieties. This is the best in the Arab regions, grown in the Arab regions, especially in Saudi Arabia. Then we tested glucose content, inverted sugars, fructose, total antioxidant, TSS content, flavonoid content, nutritional contents, and DNA isolations. The same method we use. Agar gel solutions and gel electrophoresis, the same methods. DNA weight by the electric balance using the Eppendorf tubes, electrophoresis, micropipette gel tray and the pumps. Different agar gel solutions, buffer solution, ethylium bromide. This is the most Eppendorf tubes. This is the main materials. Then methods, agar gel solutions. Ethylium bromides and then gel electrophoresis. The gels were stirred for 5 minutes in the ethylium bromide and the stains having water to 2 minutes. Then the fragments were migrated rapidly into the gel matrix based on size. So then the results, discussions and results, different results. We found conclusion. It can be concluded that our Ajwa Madina contain better nutrient antioxidant flavonoid content Ajwa Hiles and Ajwa Qasim. Ajwa Al Qasim, this is the region name, showed bigger size compared to the others cultivated trees. However, Ajwa Hile exhibits better fructose, glucose and inverted sugar compared to the others cultivated trees. This is the different types of Ajwa dates. Collected Ajwa Madina, Ajwa Hile and Ajwa Al Qasim. So different types of Ajwa Madina, large, small, large, and Ajwa Al Qasim, Ajwa High. Different types of weights, length, and diameters. Usually this is very small. Al Qasim one is a little bit bigger size than other tools. The fructose content. Al Qasim Al High, then the Al Madina. Then inverted sugars also same, higher in Al Qasim and higher than Madina Ajwa. Nutritive for potassium, calcium and content, we found the other calcium one, Madina one is higher than Al Qasim, Madina Al Qasim one higher, Madina larger, Madina small one, and this is for Al Qasim one. hyle, alqasim, Madina, Ajwal Madina, Ajwal hyle 1, potassium content, calcium content also higher in Madina 1, and then the sodium content also higher in Madina 1. Then other flavonoid, total antioxidant, pH, and total soluble solids constants also found higher in the Madina 1. Then alqasim hyle, total antioxidant found Madina large 1. Then alqasim hyle, and pH content also higher in Madina small 1 than others, but there is no significant difference. And total soluble solids higher and high than other tools. So DNA 8 found Modena 1 is higher than other tools, alqasim and Modena. This is the DNA bands, DNA segments and different as well tests. This was the small one, the large one. This is the alqasim. This is the high, this is standard. Chapter 9, we can see here carbohydrate, antioxidant, nutrient, and the DNA quantification of olive treated with alkaline and the acidic solvent. So this is the olive. In short, we can discuss this one. This is olive fruits also. Most important contents in the roles of fruits crops, especially Middle East and African countries, in the Mediterranean regions. Saudi Arabia produces 20% of olives in the world demand, so it gives the important roles in nutritional and medicinal value, and also this is very most important for the studies showed that the olives or olives oil, fresh olive oil might give a potential role in decreasing threats of the different kinds of cancer, especially colon, breast, ovarian, prostate cancer. So it has been stated that 1031 having ovarian cancer as well as 2411 lacking of cancer out of the approximately 3500 Italian women. So this is the just research data. Then we tested the biochemical concept glucose, input sugars, fructose, and the DNA isolations by gel electrophoresis. We did this total antioxidant content, TSS, PS, flavonoid content, total phenolic contents, nutrition value, and DNA isolations. The same method we use. Agarose gel solid and ethylene bromide and gel electrophoresis method. The results we can see here: different types of solvents, water, sodium hydroxide, vinegar, the vinegar plus sodium hydroxide plus hot water. So you can see here all treated fruits higher carbohydrate contents than the control. So here also it is higher in the all treated fruits than the control. And also different types of the flavonoid and total antioxidant. Here this is different types of minerals, potassium, calcium, nitrate, sodium, and here different types of pH, TSS. Here we can see flavonoid and total antioxidant. And control higher in the this is called the sodium hydroxide, vinegar, and then others contents. Then the control water, DNA yield also. The higher in the different treated rate then it is almost same. No water, DNA yield almost same. Control and others. When you use the vinegar, the list the lowest the vinegar treated olives. And here also actually DNA yield, you can see different types of yields. Here also or sage, so different types of the 1, 2, 3, 4, 5, 6. All the different treatments. So here different types of So the daily bands, characters, water contents, and the different treatments. Daily bands here, we cannot. Yeah. So you can see the different brands. So here it was published this book in the Antioxidant Fruits and Vegetables, Carbohydrate and Proteins, and Food and Human Nutrition, Biochemical and Phytochemical Compositions. In German, I published this book, Antioxidant in Fruits and Vegetables. This is most important books on the student of nutrition science or nutrition biotechnology, or those who are studying with nutrition. So this is almost today's topic. You can see here. So this is most important topic: antioxidant in fruits and vegetables, carbohydrate and proteins, and also others. So thank you very much for listening, watching, and subscribing our channel. And if you subscribe and stay with us, you will get the updated and the very innovative research data and lecture in future. Thank you again.